Hi, my name is Tiana Cushado. I do research in Dr. Kwangwan Liu's lab, and today I will be presenting our research called Characterizing Genetic Mechanisms for Measuring Day Length in Neurospora crassa. So first, here's a brief introduction. Photoperiodism is the response of an organism to a change in day length, and circadian rhythms regulate 24-hour cycles within an organism. There are two theories that explain the possible relationship between photoperiodism and the circadian clock. The first one is called the external coincidence hypothesis, which states that light is needed to regulate the circadian clock so it can respond to seasonal changes. The second one is called the internal coincidence hypothesis, which states that internal oscillators regulate the circadian clock, while the effect of light on the circadian clock is very small. The goal of our study is to determine the genetic mechanism that Neurospora crassa uses to measure day length. To do this, we used a protoparesthesia assay, or PPA, to determine the signals that need to be sent to form protoparesthesia. And protoparesthesia is a female sexual reproductive structure in Neurospora. We hypothesized that the ability to measure day length is a multi-gene trait, so quantitative trait loci, or QTL analysis, will lead us to identify genetic elements for photoperiodism. We also hypothesized that genes that are known to be involved in the circadian rhythm play a role in photoperiodism. And here's an overview of the protoparesthesia assay. First, we grew back 91 strains of wild-type Neurospora from the N6 population. Then each strain went to 3 different photoperiods for 12 to 14 days. The photoperiods were long day, which is 16 hours of light and 8 hours of dark, short day, which is 8 hours of light and 16 hours of dark, and equinox, which is 12 hours of light, 12 hours of dark. Then 4 images were taken of each plate and random spots at each of the corners, and then the amount of protoparesthesia was counted for each image. This shows the strain N681 in different photoperiods, and it shows that different amounts of protoparesthesia are produced between different photoperiods. Cohen's d is a value that shows the difference in protoparesthesia production between short day and long day using equinox as the control. We are using this value as a measure of an organism's ability to measure day length because we hypothesized that the Cohen's d value for the number of protoparesthesia produced would measure how effectively Neurospora can distinguish between photoperiods. And then here I have the equation I used to calculate Cohen's d. Next, we performed a QTL analysis, which uses genotypic and phenotypic data to find significant QTLs. A significant QTL is a chromosome that likely has a target gene somewhere on it. So this diagram shows that chromosome 5 is a major QTL, and this diagram shows that different genotypes can distinguish between different photoperiods. Next, we performed the protoparesthesia assay on 21 knockout mutants in the target region on chromosome 5, which was our major QTL. So this graph shows the raw data for the mutants on chromosome 5. The x-axis shows the different mutant strains, their FGSC numbers, and the y-axis is the average amount of protoparesthesia produced between the different photoperiods. This graph shows the Cohen's d values of the mutants in the long day condition. Using the Cohen's d graph, we determined that FGSC 17264 is a candidate gene on chromosome 5 because it has the highest Cohen's d effect size compared to the wild type. We also performed the protoparesthesia assay on 16 knockout mutants on chromosome 1. And here's the raw data for chromosome 1. And here's the graph of the Cohen's d values in the long day condition. From this Cohen's d graph, we determined that the FGSC 11617 is a candidate gene on chromosome 1 because it has the highest effect size and is also going in the opposite direction than the wild type effect size. Lastly, we performed the protoparesthesia assay on 10 clock/photoreceptor mutants. Here's the diagram of the raw data for these mutants, and here is the diagram of the Cohen's d value for these mutants in the long day condition. From the Cohen's d graph, we determined that FGSC 12467 is a candidate gene for the clock/photoreceptor mutants because it has the highest effect size, and it's also going in the opposite direction from the wild type effect size. In conclusion, we identified a major QTL on chromosome 5 and performed the protoparesthesia assay on 21 target region mutants on chromosome 5, 16 knockout mutants on chromosome 1, and 10 clock/photoreceptor mutants. We also determined that chromosome 5's candidate gene was FGSC 17264, chromosome 1's candidate gene was FGSC 11617, and the clock/photoreceptor mutant's candidate gene was FGSC 12467. I also wanted to say that this is preliminary data and we still need to repeat our experiments to verify our data. In the future, we will finish our data for the mutants in the short day condition, repeat the protoparesthesia experiments on significant mutants, and perform experiments on the candidate genes. Here are my references. And we also wanted to thank the members of the Lee Lab, which are Ningwan Zhu, Harjit Kiara, Zainab Rizvi, and Hosen Arman. And also thank you to the Mark U Star program for funding.